All right, well, I think we're going to get started. Good morning, everyone. My name is Helen Parsons. I am the co-leader of the SPECS program here in the Cancer Center. And today I have the pleasure of introducing Dr. Beshay Zordosky, who is an associate professor in the Department of Experimental and Clinical Pharmacology at the university. He completed his PhD from the University of Alberta in Canada in pharmaceutical sciences, and then went on to complete a postdoctoral fellowship also from the University of Alberta in cardiovascular research. And today, he's going to present some of his research, which is currently focused in the area of cardio-oncology, which has really emerged as a novel scientific area, looking at the ways to mitigate the cardiovascular effects of anti-cancer agents. And we know just with the growing number of cancer survivors in the United States that this is an important problem that will only continue to expand in prominence as we continue to have more cancer survivors throughout the country and the world. So today, please join me in thanking Dr. Zordosky for presenting his work, which will be on the topic of therapy-induced immunosenescence. Welcome.
Thank you. Good morning. Thank you for the introduction. So as you just heard, I have background in pharmaceutical sciences and cardiovascular research, but I'm now presenting a new focus of my lab, which is immunosenescence. So by no means I am an immunologist, so I'm collaborating with immunologists to make me understand what I'm presenting now. So, and I'm also having my notebook and pen for any comments or suggestions to improve this work. So I'm happy to listen to you, but I thought to share with you my most recent research and also so that I can get feedback from you, and most of the data that I will be presenting is not published. So hopefully this will be a nice discussion and conversation with you. So as a matter of introduction about cancer survivorship is a very good problem to have. The 5-year survival rate of cancer patients has remarkably increased, and now it reached more than 90% in some childhood cancers, breast cancers, prostate cancers, and this is thanks to most of you guys who are oncologists, cancer researchers. You advanced the diagnostics, research therapeutics, and all of these good things that culminated into the increase in survivorship. This translates into more than 15 million cancer survivors in the United States, and this number is expected to increase. And this 15 million actually was in 2016. Now it's approaching 20 million, and it's expected to be 26 million in 2040. So this is a huge population of cancer survivorship among all age groups, from the very young to the aged population. We have representation from like cancer survivors in all these age groups. This is a cause for celebration. This is a good problem to have, but this also comes with a cost, the cost of increasing health complications in these survivors, and nearly 85% of cancer survivors have one or more chronic health conditions in their lifespan. Generally, cancer survivors appear to be older than their stated age by as much as 20 years. And we will look into how biologically they are also more aged than their age-matched groups. So I will focus more into childhood cancer survivorship as my area of research, and also because if you think about children who survive their cancer, they have so many years to live. So if we can prevent these health complications, this one will be one of the most population that will benefit from this kind of research, along with like breast cancer also may happen in women in their like middle age as well. So they have so many decades to live, so that we hopefully with survivorship research, we can improve their quality and the quantity of living. So if you see here about childhood cancer survivors, this is the cumulative incidence of one chronic health conditions as compared to controls who did not have cancer. So this is a very big difference between the rates of health complications in survivors as compared to control. And this did improve a little bit with improvement in treatment. So this is like decades in the 70s and the 80s and the 90s with improved treatments. There is a little bit of side effects, like lower side effects coming out of these medications each decade, but still it is very, very high. And this is the rates of more than 2 health complications as compared to control. And this is 20 years time since diagnosis. If you imagine childhood cancer survivor, 20 years post-diagnosis means that these survivors are now in their 30th. So in like 30s, you're not expected to have this huge burden of a health complication. So there's something going on in these survivors. What are the diseases that's known to be higher in prevalence in childhood cancer survivors? Malignant neoplasm, cardiovascular diseases, hypertension, a little bit of diabetes. Obesity is the same, no much difference in obesity, but strongly peripherality and virility. These like conditions, peripherality and virility, we hear about them in the normal population only like in 60s, 70s, and 80s. Like to be really an aged, frail person, this is like in your 80s. But now you see that like 10% of childhood cancer survivors, probably in their like 40s and 50s, have this virility. So all of these are age-associated complications in childhood cancer survivors, and this strengthen the hypothesis that childhood cancer survivors develop a more aged phenotype than the controls. Another important and much less studied problems in childhood cancer survivors and also in other types of cancer survivors is immunosenescence. So immunosenescence is well characterized in the context of natural aging. So you'll find a lot about like immunosenescence when people reach their 70s or get more aged, they develop immunosenescence. This is very well known in natural aging, but much less studied in the context of cancer survivorship. So what are the features of immunosenescence? 3 important features. The first one is increased susceptibility to infections and infection-related complications. Second is a chronic low-grade inflammation, or some scientists call it inflammaging. So inflammation that's associated with aging. And third is age-associated changes in immune cell subpopulations. So these are the 3 features that we want to focus on. Are these features present in childhood cancer survivors? During the past decade, there are more and more clinical research reports showing that these features are present in childhood cancer survivors, as we will discuss now. So first, increased risk of infection and infection-related complications. This is the cumulative incidence of these types of infection in childhood cancer survivors. And as you see here, there is like within 35 years since diagnosis, almost 50% will get a serious infection or infection-related complication. And this is the mortality from infections, which is quite rare in the general population at this age, but it's increased dramatically in childhood cancer survivors. What about the incidence rate ratio? So this is the absolute rates in these survivors, how these compare to the general population. You see here, this is the rate in survivors, and this is the rate in the controls. And the incidence rate ratio is 5 times, and this is 5 years from diagnosis. So childhood cancer survivors are 5 times more likely to get a serious infection-related complication when compared to control or comparative subjects. And this is distinction here is very important. This is 5 years since diagnosis. So we are not talking about the known immunosuppressive effects of cancer treatment. So everyone knows that if a patient is receiving chemo or radiation, their immune system is suppressed during the period of treatment. So this is a very well-known and well-studied fact. But we're talking now about after finishing their treatment, and this is 5 years post-diagnosis. So currently they're not taking any chemotherapeutic drugs, and most probably, if you do the CBC or complete blood count in these survivors, they look normal. So there's no neutropenia, there is nothing that's show a severe phenotype in these survivors. Nevertheless, they still have this increased susceptibility to infection. And this happens in almost all types of infection. 5 to 10 years, this is the highest percentage of incidence rates in these survivors. Still, even 10 years post-diagnosis, there is still higher prevalence of infection-related complications in survivors. And this is bacterial infection, fungal infection, maybe not in viral. After 10 years, they are almost as prevalent as the controls. Infectious diseases have also been one of the most diseases that bring these survivors to the hospital, unfortunately. So this is the rates of hospitalization from different diseases in survivors as compared to controls. And as you see here, it's like relative risk is 1.8 in survivors as compared to the control, similar to like malignant neoplasm and diseases of blood and blood-forming organs. So this is a real problem, although it is much less studied. So this is, hopefully I convinced you with this report that there is the first feature of immunosenescence is present in childhood cancer survivor, which is the increased susceptibility to infection and infection-related complication. The second feature is inflammaging. And many reports also showing higher inflammation in childhood cancer survivors. This one was a comprehensive one that measured 33 cytokines in serum of acute lymphoblastic leukemia survivors as compared to the control. And the red means higher, and you can appreciate the control group is mostly green and the survivor group is mostly red, meaning that most of these cytokines are much higher in survivors than in control. Specific cytokines measured here in survivors as compared to control, and the elderly and HIV positive patients who also show some degree of inflammaging. As you see here, interleukin-6 is higher in survivors than in control. High sensitivity C-reactive protein is higher in survivor than in control, and much higher in the elderly. But survivors are moving toward the elderly population. The third feature of immunosenescence is changes in immune cell subpopulations. And this also showed us, this study showed us that there is change in percentage of CD4 positive cell activation in survivors, CD8 positive T cell activation, natural killer activation, and macrophage activation markers in survivors. So all of these show us that there are changes in immune cell subpopulation in childhood cancer survivors. So in this review, they put together a diagram demonstrating the immunosenescence in survivors. So cancer diagnosis, they receive chemotherapy or radiation. When they are adult survivor, there is inflammation, oxidative stress, and both of them interplay with immunosenescence. Still, it's under research that which is leading to which. So immunosenescence lead to oxidative stress and vice versa, and also inflammation lead to immunosenescence and vice versa. These are some of the changes observed in immune cell subpopulations, changes in T cell, B cells, and naive T cells, central memory, and effective memory T cells. So one area of research in my lab and in the university is cellular senescence, and we'll see how we can link cellular senescence to immunosenescence. So what is cellular senescence? Cellular senescence is a state of cell cycle arrest that's essential component of cell response to cancer treatments. Almost all cancer treatments cause some sort of cellular senescence. Persistent senescent cells disrupt tissue homeostasis and drive aging and age-related diseases. And there is a whole a list of conditions that have been shown, at least in preclinical studies, that if you remove senescent cells, these conditions improve, including cardiovascular diseases, musculoskeletal diseases, and a lot of other age-related pathologies. Although distinct, immunosenescence and cellular senescence share common features. Most importantly and most obvious is the inflammaging or the increase in inflammatory cytokines, which are known to be higher in senescent cells and also in immunosenescence phenotype. The interplay between cellular senescence and immunosenescence is being increasingly recognized in natural aging, but it's still understudied in the context of therapy-induced cellular senescence and therapy-induced immunosenescence, and hopefully this is the direction we are going to investigate in my lab in the coming few years. So wanted to see therapy-induced cellular senescence, how it relates to immunosenescence and vice versa. So from this review article, they showed that several factors lead to immunosenescent cell, like aging. Frequent pathogen exposure leads to increase in senescence in immune cells characterized by activation of p38 MAP kinase, mTOR pathway, increase in anti-apoptotic proteins like BCL2, SA beta-gal staining, and specific for immune cells, there are markers that increase in immunosenescent cells, most importantly, PD1, which is a marker of immune cell exhaustion. These cells then secrete SASP, senescence-associated secretory pattern, which is composed primarily of cytokines. And these are the anti-senescence therapy that can be used to alleviate this senescence. And I can add to this figure that cancer treatment, both chemotherapy and radiation, can also lead to this senescence phenotype in immune cells. How cancer therapy lead to senescence? There are several pathways. The main one is DNA damage, and DNA damage leads to activation of several pathways, culminating into the activation of p53 and p21 pathway, leading to cell cycle arrest and senescence. Also, it causes oxidative stress and telomere dysfunction, also leading to cellular senescence. When cells become senescent, they manifest these features. First, cell cycle arrest. They are no longer proliferating. They produced, you can observe senescent-associated hyperchromatin foci, so the nucleus become different if you see it under the microscope. There is a special stain called SA beta-galactosidase stain, so the cells become greenish when you stain them with this special stain. They also have the gamma H2AX marker of DNA damage, and there is some morphological changes. Mainly, they are enlarged and flattened. And importantly, they started to secrete SASP, senescence-associated secretory pattern, including interleukin-6, interleukin-1 beta, TNF-alpha, matrix metalloproteinases, MCP1, and other factors. Why this is important? Because this is how senescent cells may affect neighboring cells. So senescent cells are not only dysfunctional, but they also bad because they can affect neighboring cells by secretion of these senescent-associated secretory pattern, leading to age-related diseases. Evidence of senescence in childhood cancer survivors. As you can see here, this is the leukocyte telomere length. It is reduced in survivors, and also P16, which is a marker of senescence, it increases in survivors. Interestingly, because of these, like in clinical trial, they measure these senescence marker mostly in peripheral blood mononuclear cells, which are still immune cells. So if you see like most of the studies that showing evidence of senescence in childhood cancer survivors, they are actually showing this evidence in immune cells, signifying that these childhood cancer survivors have immunosenescence phenotype. So immunosenescence If you think about it, you link it directly to increased risk of infection and infectious-related complications. But it also have been associated with other diseases or age-related diseases, mainly secondary malignancy. So if the immune system is aged, there is lower or disrupted immune surveillance of any cancer tissues or tumors that's arising in later on in cancer survivorship. So there is an association between immunosenescence and secondary malignancy. Immune cells play an important role in the cardiovascular systems, in myocardial fibrosis, in atherosclerosis, all of this. So there is also an association between immunosenescence and cardiovascular diseases. And definitely there is also an association between immunosenescence and peripherality and ferality. Most of these studies show association, not a clear cause and effect relationship. However, in I think 3 years ago, there is a very unique study and very strong study from the Paul Robbins and Dr. Niedernover lab in the university here in IBAM. In this study, they induced the DNA damage specifically in hematopoietic cells to induce an aged immune system. So here, this ERCC, it's an enzyme that clears DNA damage. If you lower the expression of this enzyme, you increase DNA damage. So they linked this to hematopoietic cells. So now the DNA damage is happening primarily in hematopoietic cells, and senescence is only happening in hematopoietic cells. But when they do this, they found out that senescence is not only increasing in hematopoietic or lymphoid organs, but even in solid organs and non-lymphoid organs. So if you see here in these mice, which has this specific ERCC knockdown in hematopoietic cells, there is an increase in senescence markers in almost all tissues: aorta, lung, liver, pancreas, gastrointestinal tract, kidney, heart, brain, and adipose tissue. One of the hypotheses explaining that is immune system is important in clearing senescent cells. So if there is an immune senescence phenotype, senescent cells are not cleared, and then they start to accumulate at a higher rate. So this is a very direct evidence showing that immunosenescence or an aged immune system can drive senescence and aging-related diseases in other organs. Not only about markers, but about the function of these organs as well. So if you see here, ALT and AST as liver function markers, you can see that in these mice with DNA damage in the hematopoietic system, there is an increase in ALT, and this year ones are the aged mice. So these are young, but with DNA damage in their immune system, and this is the elderly, showing that there is a liver dysfunction, and this is urinary protein excretion, showing also there is a kidney dysfunction. SASP is increased, SASP factor is increased, including GDF15, interleukin-1 beta, MCP1, and TNF-alpha. Importantly, there is a decline in survival and reduction in the age, median age in this group. So all of this culminating into evidence showing that an aged immune system drive aging in the whole organism and age-related diseases. So coming into little bit into my research area, which is pharmacology. So how to get rid of these senescent cells, hopefully reduce the number of senescent cells, and then also manage or mitigate immunosenescence. So there is a new pharmacological class of drug called xenotherapeutics. Some of them are known and repurposed as xenotherapeutics, and some of them are novel therapeutics. So the novel group is called senolytic drugs, and senolytic drugs are drugs that selectively eradicate senescent cells. So senescent cells, one of the factors that makes senescent cells stick in the body and not easily cleared and not easily die is that there is an increase in the anti-apoptotic pathway in these cells, including BCL2 and PI3K AKT pathway. So these increase, make them survive, stick around, and secrete a lot of SASP factor. So the senolytic drugs selectively inhibit these anti-apoptotic pathway that are highly expressed in senescent cells, and this leads to selective apoptosis of senescent cells. So you may think that, and I'm also sharing this kind of thinking with you, if you have a senescent, like if chemotherapy induces senescent cells increases, so now I'm killing senescent cells, so I'm increasing apoptosis. And we actually found out that if you treat cells either in vitro or animals in vivo with doxorubicin chemotherapeutic agent, and then you start administering senolytic, there is an increase in apoptosis. So how this is beneficial? The theory is that this is beneficial because senescent cells are bad not only for themselves, but for the neighboring cells. So if you eradicate senescent cells, there is an initial increase in apoptosis, but then you get rid of the SASP, and hopefully other cells will be happier environment, and the tissues will be happier and will be more healthy. Another arm is the xenomorphic drug. So xenomorphic drugs, they are mostly all of them are repurposed drugs like resveratrol, metformin, and rapamycin. So these xenomorphic drugs don't kill the senescent cells, but they inhibit the SASP, which is the main bad thing secreted from these senescent cells affecting neighboring cells. So they inhibit SASP secretion. Hopefully that senescent cells will stay, but will not harm other neighboring cells. And another very interesting study from the Dr. Robbins and Dr. Niedernover group, and first authored by my immunology collaborator, Dr. Christina Kamel, that they showed that senolytic drugs mitigate immunosenescence in natural aging. So if you see here that these are young mice, that young and old mice are exposed to infection through the NME approach or natural microbial exposure approach. So they just put the mice, we all keep our mice in SPF condition, like standard pathogen-free condition, very clean environment. So they get these mice and they co-house them with pit mice or with pit mice like flooring and like mite. So they get the exposure of infection that pit mice get. And these mice are not used to that. So young mice can tolerate that, they survive, they get infection, but they overcome it and survive. Old mice, unfortunately, die when they get this kind of infection. So as you see here, it's 100% mortality rate in mice when they get this normal microbial exposure. If you treat these mice with senolytic drugs like physin and dasatinib plus quercetin, you save almost half of these mice. So this is another direct evidence showing a link between senolytics, removal of senescent cells, and immunosenescence and susceptibility to infection. Not only they save these mice life from this infection, but also reduce the P16 and P21 as markers of senescence in all these organs, and importantly, reduce SASP and inflammatory cytokines in blood, interleukin-1 beta and interleukin-6, significantly reduced by physin in these mice. So this is the young mice, this is the old mice, and this is old mice when they are treated with physin as a senolytic drug. So this is in the context of natural aging. What about in therapy-induced aging models? So we started to look into that by using a first very simple technique in endothelial cells. We wanted to induce senescence in endothelial cells using a traditional chemotherapeutic drug, doxorubicin. Doxorubicin is an anthracycline chemotherapeutic agent. It's known to be cardiotoxic. It's known to induce senescence. And although all of this toxicities is still widely used because it's very effective. Almost 50% of pediatric cancer patients still receive doxorubicin, and also breast cancer patients receive, not all of them, but breast cancer is still an option in treatment of breast cancer patients. So what we did here is a very simple experiment. We get two types of endothelial cells, HUVEX and EA cells. We treated them with increasing concentrations of doxorubicin. The maximum concentration we used here was a 0.5 micromolar, so that we induce senescence, not apoptosis. Doxorubicin induces apoptosis at 1 micromolar. So 0.5 was the highest concentration here. We removed the doxorubicin after 24 hours, and we left the cells in doxorubicin-free medium for 3 days. We measure senescence markers, cell cycle analysis, CESP gene expression, and cytokine analysis in the media. And we left them for 120 hours for SA-beta-gal analysis. As expected, doxorubicin caused robust senescence in endothelial cells as marked by a cell cycle arrest and staining of endothelial cells by the SA-beta-gal. So SA-beta-gal stains cells with like this bluish to greenish color, and almost all cells are stained with doxorubicin. And just to reach because doxorubicin causes like these cells do not proliferate after you add doxorubicin, completely stop the proliferation. So to get this density of cells, we need to seed these wells differently, and we did a lot of pilot experiments, and we reached, I think, 1:60 seeding ratio. So these are like the controlled cells need to be much less density so that they can give them a chance to proliferate. But these, like doxorubicin treated cells do not proliferate. Senescence markers increases specifically in the p53, p21 pathway. We did not see changes in the p16 pathway, and this is consistent both in vitro and in vivo. We do not in our hands, we do not get any upregulation of p16 in doxorubicin-induced senescence or aging models. Only the p21 that's increased. Cytokine secretion. These are cytokines that are measured in the medium. So these are the secreted cytokines into the media. So if you imagine these endothelial cells lining blood vessels in almost all organs in the body, and then they start secreting these interleukin-6, interleukin-8, TNF-alpha, interleukin-1 beta, and MCP-1, they will start to induce inflammation in neighboring organs as well. To use this system, to use this model, we wanted to see what is the effect of a xenomorphic drug, and we used in this experiment, we used metformin. Metformin is originally an anti-diabetic drug, and it's known to have a xenomorphic effect. So we wanted to see what is the effect of metformin in this context. So we treated cells with metformin for 24 hours, then added doxorubicin for 24 hours, removed the doxorubicin, added back metformin for the 72 or 120 hours. Metformin reduced the effect of doxorubicin on senescence markers, p53 and p21, and partially mitigated doxorubicin-induced senescence. Doxorubicin alone induced SA-beta-gal. Dox plus metformin partially reduced the SA-beta-gal staining. But more importantly is the reduction of CESP markers with metformin. As you see here, even the 2 millimolar, which was not very effective at like reducing senescence marker, but it definitely reduced the CESP secretion, which may be giving us a hint that metformin has a broader anti-inflammatory effect in this context. Another characteristics of aging or CESP or inflammaging is that not only the cells and the mice and humans have higher inflammatory burden, but they also respond more vigorously to inflammatory stimuli. So if you get like a mouse with LPS, this is a young mouse, LPS increases inflammation. Lipopolysaccharide is a bacterial cell component known to induce inflammation. This is inflammation that's induced in young mice. As you see in old mice, there is an exaggerated response to LPS, both on all these markers. And this is called hyperinflammation or hyperinflammatory response to LPS. This is also known in natural aging. So does this happen in doxorubicin-induced senescence, and what is the effect of metformin? So we did the same experiment. We added metformin, then doxorubicin, then removed doxorubicin. Then we removed the media because we know that these media have the cytokines already, so we removed them, and then cultured the cells with fresh medium and metformin and LPS. So LPS now is inducing inflammation in these cells, and we want to measure the cytokine induced by LPS and what is the effect of metformin. Interestingly, there is this hyperinflammation still present in doxorubicin-induced senescence. So this is a response to LPS in proliferating control cells, and this is a response to LPS in doxorubicin-induced senescent cells. And you can appreciate the fold of difference between when cells are normal and when they are senescent. Interestingly, metformin reduced inflammatory markers in both the controlled cells and also in the hyperinflammatory cells. So interleukin-6, CXCL2, and TNF-alpha, all of them showed the same pattern of hyperinflammation with LPS, and this was brought down by metformin. Other markers specific to endothelial cells, we measured ICAM1, which is an adhesion molecule known to be increased in endothelial cell inflammation. And you see that LPS alone increased ICAM, but the increase was much higher when doxorubicin-induced senescent cells were treated by LPS. And all of this were mitigated by metformin. Mechanistically, we showed that the P65 NF-kappa B pathway may be the mediator here. We demonstrate that it increased with LPS as expected, but also in doxorubicin, it's increased with doxorubicin-induced senescence, further increased with LPS, and brought down by metformin. So one of the effectors of metformin might be the NF-kappa B pathway, and we'll be looking into this in more detail using other NF-kappa B inhibitors, which are also known to have xenomorphic effects. Then we wanted to do a mouse model for doxorubicin-induced immunosenescence to see the changes in immune cell subpopulation, which we cannot do in our simple in vitro experiment. So we used this model of very low dose of doxorubicin, 4 milligram per kilogram, once a week for three weeks. And I chose this dose because I have used them in my cardiovascular research experiments, and they are, first, they do not cause mortality at all in these mice. A problem we see in preclinical studies is when you increase the dose of doxorubicin, there is a lot of mortality, and we don't want to see this. This dose also does not cause severe morbidity. So it's a good, if you want to relate it to the clinic, it's around 40 milligram per meter square, which is a very, very low dose of doxorubicin in pediatric cancer patients. So it's very, very minimal. We administered this to mice for 3 weeks, and then we sacrificed a subset of mice at 1 week and at 5 weeks post-dox. To remind you, 1 week in a mouse life is almost a year in human life. So 5 weeks after the last dose of doxorubicin, this means that it's 5 years. So this is a long-term effect of this chemotherapeutic agent. So first, we did a simple CBC to see what are the effects on the total blood counts. As expected, at 1 week, there was a modest decrease in white blood cells, and there was a trend toward an increase in monocytes. Nothing else is changed, and this is because this dose is very, very low, and also 1 week in mouse life is almost a year in human life. So if you expect a pediatric cancer patient, after receiving treatment for 1 year, the immune system starts to reconstitute. At 5 weeks, there is almost no change in all blood counts except the same trend in increasing monocytes still there. So with the help with Dr. Christina Kamel lab and her students and postdocs, we got the spleen and also the thymus from these mice, and we did flow cytometry to show the effect of doxorubicin on immune cell subpopulation. First, we measured P21 gene expression in the spleen, and we found that doxorubicin increases the P21 gene expression, indicating or suggesting an immunosenescence phenotype. Then we measured the different cell types in the spleen. There is a slight decrease in CD45 positive cells, a decrease in natural killer cells, no change in macrophages or CCR2 positive. When we did the T helper cells or the CD4 positive cells, there is no difference in the frequency of CD4 positive, but there is an increase in the exhaustion marker PD1 in both the naive and the effector T helper cells. Similar, the cytotoxic T cells, the CD8 positive, there is no difference in the frequencies of these cells, but there is also an increase in PD1 positive cells. As a follow-up experiment, Dr. Kamel wanted to see whether this effect is happening only in vivo or can happen in vitro as well. So she isolated splenocytes from the spleen, treated them with doxorubicin in vitro, and measured these markers. She also found that doxorubicin increases PD1 in splenocytes in vitro as well. After 5 weeks, P21 gene expression is still induced in the spleen, but we found out that most of the changes have been normalized, except there is actually an increase in CD45 positive cells, as maybe a compensatory mechanism. Spleen started to regenerate, or more CD4 positive cells. But importantly, there is an increase in CCR2 positive myeloid cells in the spleen, indicating a more inflammatory immune response. No major changes in CD4 positive cells, but there is still an increase in PD1 positive cells in naive cytotoxic T cells. We're trying now to increase the dose of doxorubicin, hopefully to see more robust changes at the 5 weeks, so that we can further illustrate an effect on the immune response in these mice. But we were quite surprised to see an effect of this very low dose of doxorubicin after 5 weeks of treatment. This is the spleen. We also have very interesting data on the thymus, but I don't want, because it becomes very messy if you present it to organs. But with the thymus, the interesting thing is that at 1 week, there is very significant and remarkable thymus involution or thymus atrophy. So thymus very, very small, and this is known for doxorubicin. But to our surprise, at the 5 weeks, there is actually a hypertrophy or hyperplasia of the thymus. So it's not only came back to normal, but it actually overgrew and became more than the normal. This was also associated with a lot of changes in the immune cell subpopulation, and we're trying now to put this together to find out how these changes may have an effect on immune function and on other functions as well. So what is the effect of metformin on this? So this is a work in progress, and we did a very preliminary study when we treated mice with doxorubicin plus or minus metformin. The dose of metformin is 250 milligram per kilogram per day in drinking water. This is very clinically relevant dose. It's approaching like 2,000 milligram per day in human, which is clinically used dose of metformin. Metformin was started before doxorubicin, concurrent with doxorubicin, and for 5 weeks post-doxorubicin. We are currently doing the flow, but interestingly, even at the CBC count, there are some changes, which is very remarkable for metformin as a clinically relevant dose to increase white blood cells, reduced neutrophil count in doxorubicin-treated mice, increased lymphocyte percentage in doxorubicin-treated mice, abrogated the effect of doxorubicin on monocyte, and doxorubicin has, for two-way ANOVA, it gives you the effect of metformin. Metformin also had a significant effect on eosinophils. We expect that since these changes are happening at the whole blood CBC, we expect even more changes are happening at the subpopulation analysis by flow cytometry. As metformin has a clinical relevance in cancer patients. So you probably know that metformin in preclinical studies, especially in vitro, showed a very remarkable anti-cancer effects and anti-proliferation, and this made many clinicians excited to try metformin in cancer patients to see whether it may add to anti-cancer effect. Unfortunately, most of the studies I am aware of did not find a significant effect of metformin as an anti-cancer agent. But surprisingly, some studies showed benefit of metformin in cancer treatment beyond the tumor reduction effect. So one of these studies is this one. It's in a diffuse large beta cell lymphoma with diabetes. So these patients were not specifically treated with metformin for clinical trial purpose. They were already diabetic and they were receiving metformin for their diabetes treatment. And they found out that diabetic patients treated with metformin have better overall survival than diabetic patients without metformin. Some other studies in breast cancer patients, although they did not find an effect on the tumor, they found out that metformin significantly reduced some side effects of cancer treatments, including neutropenia, which again make us think of metformin not only for its anti-cancer effect, but maybe it protects the immune system during cancer treatment and maybe after cancer treatment in the immunosenescence phenotype observed 5 to 10 years after treatment. The role of metformin has also been highlighted with the COVID-19 epidemic, and this is another study coming from the university here from Dr. Brament, who did this interesting clinical trial in treating COVID-19 patients with metformin, and they found that cumulative incidence of long COVID is lower in patients receiving metformin as a treatment or as preventive treatment during their COVID. So the long COVID, there was a trend toward better hospitalization and mortality, but the main effect was actually in the cumulative incidence of long COVID. And when I looked into this, and I should have known this as I'm College of Pharmacy, metformin is a very interesting molecule because it was originally developed in the 60s as anti-infective drug. So it was given as anti-infective drug. They didn't find it kills bacteria or anything, but because it lowers the inflammation, some patients were better with metformin than without it. But then they discovered it's anti-diabetic drug, so it's repurposed as anti-diabetic drug and then became one of the most commonly used anti-diabetic drugs. So we can kind now of like trying to repurpose metformin back into its role in infection. So in conclusions, there is a vicious cycle between cellular senescence and immunosenescence. So if you induce cellular senescence in immune cells, this leads to immunosenescence. When immunosenescence happens, immune surveillance is reduced and senescent cells are not cleared, so that more burden of senescence happens. Doxorubicin induces features of immunosenescence in vitro and in vivo. We measured the inflammaging and we measured the changes in immune cell subpopulations. We still have to get a good model to show whether or not doxorubicin also increases the chances for infection-related complications and mortality. Metformin as a xenomorphic drug mitigated DOX-induced inflammaging in vitro and may abrogate DOX-induced immunosenescence in vivo based on our preliminary finding of these changes in CBC. So what questions are unanswered? What is the effect of doxorubicin plus or minus xenotherapeutics like metformin and senolytic drugs on infectious-related complications? And this is ongoing in collaboration with the Camille Lab. And another important question that I'm trying to bring this into my own area now, what are the implications of dox-induced immunosenescence beyond increased risk for infection? And primarily, I'm interested to see what is the effect of dox-induced immunosenescence on cardiovascular complications. And then I am fortunate that I received the Masonic Cancer Center pre-R01 to answer this question. And our main hypothesis with the increase in CCR2 positive cells, it's known to be implicated in cardiovascular biology. So we think that there is a link between doxorubicin immunosenescence and cardiovascular diseases. Another area that I would be interested in either doing it myself or if someone is interested in seeing that, we can talk about it, is secondary malignancy. What about childhood cancer survivors who are known to have higher rates of secondary malignancy? Can this be related to the immunosenescence phenotype and the immune function is lower in these cancer survivors so they cannot, like immune surveillance is compromised and this give chance to a secondary malignancy? I would like to highlight the grant mechanism that made all this research possible. This was funded by the Childhood Cancer Research Fund Survivorship Research Award that I received a year ago. And this grant, I am very fortunate for it because in their letter of intent, they said that they want something that collaborative, innovative, creative, beyond your area or comfort zone. So I had this idea brewing in my mind, but I don't have any money to do it. And this mechanism was perfect for that because we didn't have any preliminary data. The collaboration with Dr. Christina Camille was, we were talking to each other, but there was no formal collaboration between us. So this came just in time. We applied and we got it. So thank you very much for the Childhood Cancer Survivor program. And these are the lab members who worked on this. Definitely my lab manager, Marian Grant, and my former postdoc, Beno George. And when the in vitro models was my former graduate student, Ibrahim Abdigawad, and former postdoc, Kevin Agostinishi, and other grant funding. And as you see, my grant is actually from the NHLBI. So up till now, I am a cardiovascular researcher. Thank you very much for listening. I'm happy to answer your questions.
I have microphones for questions.
Hi, I was just wondering if you could speak to whether or not, what you would hypothesize would happen if you removed a patient off of the metformin after treatment, whether or not you would have cured and removed that kind of cycle that you had spoken about forming, or whether or not there would be kind of the recreation of that environment.
That's a very good question. We're currently doing different experiments to see, so to make sure that we got something, we had a very broad umbrella, a very broad net. We used metformin with doxorubicin and after doxorubicin, but your question is very important. And I would add to it another thing. So doxorubicin with metformin is important to see and stop it after, but also metformin after doxorubicin. So what about children who already received their treatment and now they're in their adulthood and start to have this inflammation? If we treat them now with metformin, what's going to happen? So we're designing now an experiment to answer this question. So plus the arm that have metformin with and after, we'll have two other arms, metformin only with doxorubicin stopped, and another arm metformin just after doxorubicin. Thank you for the question.
Hi, that was a great talk, and it points out there are a lot of ways to damage DNA in cancer therapy. So the question, I guess, is how important do you think this is a mechanism for doxorubicin as opposed to other drugs? You know, kids get a lot of drugs. Some of them are anthracyclines, as you mentioned, in breast cancer, trying to not give anthracyclines as much as possible. and give antitubulins. So does it make a difference if you use doxorubin than say paclitaxel? And a kind of related question is, how much do you worry about an anti-senolytic could affect the therapeutic effect of the drug? You know, as you know, the only drug that ever got approved for doxorubicin-reduced toxicity is Zenocard. Nobody uses it because the data suggests it makes the doxorubicin work less well on the tumor. So I'm just thinking about, do you want to build tumor models into your model of senescence?
Great, thank you for the question. So the first question is, we think that most chemotherapeutic agents would do the same, and also radiation if it's directed toward like the bone marrow or the immune organs. So all of them will lead to p53 and p21 activation and immunosenescence. Even more than that, there is evidence that specifically in childhood cancer patients, pediatric cancer patients with lymphoma and leukemia, they show signs and features of immunosenescence even before they start the treatment. So this is another issue which we're trying to tease apart with animal models. What is the effect of chemotherapy versus the effect of cancer itself? So even cancer itself can induce immunosenescence. So I think this is a common theme in pediatric cancer patients with different agents of chemotherapeutic drugs, and may also be with cancer itself. For your second question, we have been doing very good collaboration with Dr. David Largispada, advising us on a lot of tumor model. I have a lot of data about tumor and tumor-bearing mice. So specifically for metformin, I can, at least in the lymphoma model that we have, we tried it with doxorubicin. It does not reduce the effect of doxorubicin. Unfortunately, it does not potentiate it, as some labs were showing that metformin has anti-cancer effect, but it did not reduce the chemotherapeutic effect of doxorubicin. But we tried other xenomorphic drugs like losmapimod, which is a p38 inhibitor, and it does reduce the chemotherapeutic effect of doxorubicin. So definitely building tumor-bearing mouse experiments along with this is very important with the xenomorphic approach, because if you want to administer something with doxorubicin, this is a hurdle that you need to answer. And in my opinion, as you specifically mentioned about the cardioprotective agent dexrazoxane, although it showed protective effects, still oncologists are hesitant to use because they don't want to mess up with whatever regimen they are giving. So the senolytic and xenomorphic approach, if we can find out that they protect even after doxorubicin treatment, in my opinion, this will be a better approach. Because imagine now a childhood cancer survivor or breast cancer survivor treated from their cancer, now they're cancer-free. Now you are protecting other organs by administering the chemotherapeutics after the treatment is done. Because to be honest, if you want a translational path, it's better not to mess with the cancer treatment. Don't give anything with the cancer treatment to appeal for oncologists to adopt your strategy. But we're also doing that in tumor-bearing mouse model. For senolytics, it's maybe even better for cancer because it's a two-hit. You induce senescence in tumors by the cancer treatment, and then you administer the senolytic, and then the senolytic will even kill the cancer cells which were senescent. So with the senolytic, it may even be more advantageous to give this a two-hit cancer-inducing senescent agent plus a senolytic approach. This can help the tumor and may relieve some of the long-term complications as well. But other treatments were, definitely senescence is very important in cancer control. So we don't want to get rid of senescence while the cancer patients are being treated.
Do we have any other questions? All right, let's say thank you one more time.
Thank you.